Hello and Assalamualaikum everyone. In this video, we will learn about meiosis, which is the subtopic under cell division. But before that, we are from Group C and this is our group mates. Let's start by definition of cell division. What is cell division? Cell division is the process by which a single parent cell split to form new cell. There are two types of cell division, mitosis and meiosis. But in this video, we only focus on the topic of meiosis. Let's go through. What is meiosis? Meiosis is a type of cell division that necessary for sexual reproduction to occur, as it results in the formation of gametes, sperms and eggs. Meiosis is an exceptional phenomenon which produces 4 daughter cells, each with half the number of chromosomes. Next, the learning objective that we want to highlight in this video are to state that meiosis involves prophase, metaphase, anaphase, and telophase, which 4 genetically different haploid cells produce, and to understand how the crossover of homologous chromosomes increase genetic variation.
Meiosis 1. There are 5 steps for meiosis 1. Firstly, interphase. Preceding meiosis, which is DNA replicates. Next, prophase 1, homologous chromosome condense, pair up, and swag segment. Spindle microtubule attached to chromosome pair are aligned midway between spindle poles. Metaphase 1, the homologous chromosome pairs are aligned midway between spindle poles. In anaphase 1, the homologous chromosomes separate and begin heading toward to the spindle poles. In telophase 1, 2 clusters of chromosomes reach the spindle pole. A new nuclear envelope forms around each cluster, so 2 haploid nuclei form.
Meiosis 2. Meiosis 2 is the second meiotic division and usually involves occasional segregation or separation of sister chromatic. Mechanically, the process is similar to mitosis, but for its genetic results are fundamentally different. The 4 main steps of meiosis 2 are prophase 2, metaphase 2, anaphase 2, and telophase 2. Next, we will see the first stage of meiosis 2, which is prophase 2. In this stage, the chromosome condense and the spindle microtubules attached to its sister chromatic as the nuclear envelope break up. The process will move to the next stage, that is metaphase 2. In this stage, the still duplicated chromosomes are aligned midway between the poles of the spindle. Next is anaphase 2. In this stage, all sister chromatics separate and the now unduplicated chromosome head to the spindle pole. The last stage for the process of meiosis 2 is telophase 2. In this stage, the 2 new sets of non-identical cells are generated from each divided cell. The nucleus starts to form around each new cell.
The differences between meiosis 1 and meiosis 2. For meiosis 1, chromosome arranged at the equator of the cell in homologous pair. Meanwhile, in meiosis 2, chromosome line up at the equator of the cell. Whole chromosome move to opposite pole of the cell in meiosis 1. Meanwhile, in meiosis 2, daughter chromosome or chromatids move to opposite pole of the cell.
Now we are going to continue with ethical issue in this subtopic about genetic screening and discrimination. As our understanding genetic improve, there is a potential for a misuse of genetic information obtained from meiotic cell. Genetic discrimination could occur when individuals are treated unfairly based on their genetic predisposition, leading to issues such as denial of insurance coverage or employment discrimination. So these are not ethical to be practiced because it will be discriminating the affected people. In conclusion, meiosis is a specialized form of cell division that occurs in sexually reproduction organisms. It involves 2 successive divisions, known as meiosis 1 and meiosis 2, resulting in the formation of 4 haploid cells from a diploid parent cell. The main purpose of meiosis is to generate genetic diversity and ensure the proper distribution of chromosomes during gamete formation.
So that's all from us. Thank you.